All right, welcome. Today, we're diving into a story that honestly feels like it's pulled right from a sci-fi movie. We're talking about scientists who are solving a river's deepest secrets, not with nets or boats, but with just a few drops of water. So let me just pose a question. Imagine you're a scientist. Your job is to catalog every single fish species living in a massive 400-kilometer-long river. It's remote, it's murky, it's huge. I mean, how on earth would you even start a project like that? Well, here's the brilliant, kind of mind-bending answer. You don't. You don't try to catch all the fish. You let the river itself tell you who's living there. Because it turns out, every single creature leaves behind a tiny, invisible calling card just floating in the current. And the key to this whole thing is a revolutionary tool that is absolutely changing the game for conservation. It's called environmental DNA, or eDNA for short. Okay, the best way to think about it is like this. As a fish swims through the water, it's constantly shedding tiny bits of itself. Skin cells, scales, all that stuff. Well, that material contains its own unique DNA. So that DNA just floats in the water, kind of like a cloud of invisible glitter. And what scientists can do now is just scoop up some of that water, find those genetic fingerprints, and figure out exactly who left them behind. And the process, when you break it down, is surprisingly simple. First, you just collect a water sample. Then you run it through a super fine filter that traps all that DNA. Back in the lab, machines sequence those little DNA fragments. And finally, a computer matches them against a huge genetic library to get a complete list of species. It's basically CSI for an entire ecosystem. So this isn't just some cool lab trick. Let's look at a real-world case where this exact technology was put to the ultimate test. We're heading to the Paranaíba River in Brazil, where the stakes really couldn't have been any higher. So here's the situation. You've got this 400-kilometer stretch of river, one of the last big, free-flowing sections left in the whole area. It's a superhighway for migratory fish. But a new hydroelectric dam was being planned, which would have just chopped it up forever. And the biggest problem? Nobody really knew how important this part of the river was, because its fish life was a total mystery. So to survey this massive river, how much water do you think the scientists needed? You're probably picturing huge tanks, right? Nope. At each site, they collected an average of just 360 milliliters. That's less than a can of soda to map out the life in 400 kilometers of river. Talk about an efficient approach. All right, so the team collected their tiny samples, ran the analyses, and they basically waited for the river to tell its story. And what a story it was. Get this. From those few little water samples, the eDNA analysis identified 74 different fish taxa. That means 74 distinct species or groups of species. This was way higher than anyone expected. It was the first clue that this river's biodiversity was far richer than anyone had ever guessed. And the data also revealed this really crucial ecological pattern. So the upstream sites, they had a pretty consistent amount of fish life, about 18 different kinds. But the one site located downstream of an existing dam, that number exploded to 40 distinct taxa. This was huge. It proved the method was sensitive enough to detect real ecological patterns, confirming that the technique was actually working. But here, this is where the power of eDNA really just becomes crystal clear. A previous survey, one that used traditional nets and gear inside the reservoir of that same dam, it had only found 12 species. This new eDNA survey, just downstream, found 40. See, it's not just that it's easier, it's revealing a much, much more complete picture of what's actually living in the river. I mean, the researchers themselves said it best. They concluded that this approach gave them a, and I'm quoting here, rapid, robust snapshot of the river's fish community. In other words, it was fast, it was reliable, and it gave them exactly the kind of solid data you need for urgent decisions. And it gets even better. The DNA detectives didn't just find general patterns, they found specific high-value targets. From just a single water sample, they detected the DNA of the pintado, a large migratory catfish that's actually a threatened species. This is a fish that a net could easily miss, but its invisible genetic fingerprint gave it away. So we have this incredible data, a rich, detailed picture of a vibrant, vital ecosystem. But what does it all mean? Well, this is where the story turns from just fascinating science into a real-world conservation victory. And here it is, the verdict. The scientific team states it directly in their paper. The rapid biodiversity assessment they produced using eDNA was instrumental, that's their word, in justifying the government's decision to deny the construction license for the new dam. Those tiny water samples helped save 400 kilometers of free-flowing river. Just amazing. So when you put it all together, it's pretty easy to see why this is such a total game changer. eDNA is faster and more affordable. It can find those rare and hidden species we might otherwise miss completely. And maybe most important of all, it provides the clear, powerful evidence needed to make informed decisions that can actually protect our planet. And that really just leaves us with one final thought. If a few milliliters of water can help save an entire river in Brazil, just imagine what other secrets are hiding out there in our planet's oceans, lakes, and streams, just waiting for the right detective to come along and test the waters.